All right, this video is going to cover it should say chapter 14 on your page 91 in your binder. So remember, when you're watching an Edpuzzle, there are questions that pop up. So you're going to want to pay attention to what's going on in the video so you can answer those questions. It's just an accountability thing. And then make sure you're filling out your binder notes at the same time. The reason why we have to do this a little bit more frequently right now is because we have a deadline to hit a certain point in the curriculum before Thanksgiving break. We've got to start breeding our fruit flies. So I'm trying to get us on track the best I can. So we're on page 91, and this chapter is about the synthesis of proteins from DNA. So the last chapter we did was just DNA replication. So that's just copying DNA before a cell divides. What is DNA actually for? Like, what does it actually code for? And that's proteins. So gene expression is basically DNA being transcribed into RNA and then RNA being translated into proteins. Skip where it says old idea, skip newer idea, write down most accurate. Just skip old, skip newer, write down most accurate, because this is the one that's correct. So the idea is one gene codes for one RNA, which gets translated into one polypeptide. So one gene to one RNA to one polypeptide. Pause the video anytime you're still writing. I am going to move on. We've already written this down before. The flow of genetic information is DNA to RNA to protein. Transcription is DNA to RNA. Translation is RNA to protein. All right, so that's the flow. DNA to RNA to protein. Transcription is DNA to RNA. Translation is RNA to protein. The site of translation, the site of protein synthesis, is going to be at a ribosome, either a bound ribosome or a free ribosome. So go ahead and pause if you need to write this down. Something that's important to know is where transcription and translation happens. So in a prokaryote bacteria, transcription is going, and translation are just going to happen on the cytosol. On your notes, though, it asks what is transcription and where is the site in eukaryotes? So transcription is DNA to RNA, and in eukaryotes, that happens in the nucleus. Translation is the RNA being translated into proteins, and in eukaryotes, that happens outside of the nucleus at the ribosomes. So it's important to know transcription occurs in eukaryotes in the nucleus. Translation occurs in eukaryotes in the cytosol, or outside of the nucleus, wherever the ribosome may be. In prokaryotes, though, it all happens just in the cytosol because they don't have a nucleus, right? All right, so one gene equals one RNA molecule. Let's talk about the differences between DNA and RNA. So you can make a Venn diagram. There's not a lot of room there. I'm sorry, I should have left more room on that page. So on page 91, you'll need to pause the video and write down the differences. So they're both nucleic acids. That's what they have in common. All right. DNA is double-stranded. RNA is single-stranded. The sugar in DNA is deoxyribose. The sugar for RNA is ribose. DNA has a nitrogenous base called thymine, while RNA has uracil. DNA is a template for proteins, but RNA, there's a lot of different roles for it. And I'm going to tell you some of the different types of RNA in just a second. So pause the video and write this down because I'm moving on. The different types of RNA I have listed here, lots of different roles for RNA. So pre-mRNA is what's going to be newly transcribed in a eukaryote. So pre-mRNA, that's basically RNA, but it's going to end up being edited. In a eukaryote, mRNA is the edited version. tRNAs are actually going to assist in translating mRNA. So they're going to bring in amino acids, and we'll talk about that a little later, and I have a game for you to play with that. rRNA is the RNA that makes up 60% of ribosomes. Ribosomes are made up of rRNA and proteins, and they are the site of protein synthesis. snRNA small nuclear RNAs. They're part of these things called spliceosomes, and I'll show you what those are in a little while. And then skip number 6. That one's not on there. Don't worry about that one. The last blank down there is a ribozyme. It's an RNA that functions as an enzyme. So you're filling in all of these on page 91, except number 6. Just don't worry about srpRNA. But I mean, there's other types that we aren't going to talk about. So RNA has a lot of different roles. So make sure you write this down, pause the video if you need to, get it written down. And then I'm going to move on to the steps of transcription. So for each gene, one of the DNA strands is the template strand. It's the one that's going to be transcribed. The mRNA is going to be complementary to the template strand. So if you see right here, look at my cursor, the top strand right here is the template strand of DNA that's going to be transcribed. RNA, the red strand right here, is complementary to that. So in DNA, A pairs with T, but with RNA, there are no T's. So anytime you would have put a T with DNA, you're going to have a U with RNA. So A pairs with U, C pairs with G, C pairs with G, A pairs with U, A to U, A to U, C to G, C to G, G to C, A to U, G to C, T to A. Now I know y'all did that in cell bio, so I'm not gonna we'll do some practice for sure. Oh yeah, you'll definitely get some practice on that. But just know, template strand, then you have a complementary RNA. RNA does not have T's, it has U's instead. And then translation is where the codons, they're read in triplets. So the RNA is read in 3s, will translate into a string of amino acids. Okay, so the mRNA triplets, the codons are going to code for amino acids. You should, or next time I see you, I will give you a codon chart, but we're not going to do, oh, you have a codon chart on page 101. We're not going to do translation today, though. We're just on transcription. So I'm going to try to get through the steps of transcription, and then I'll end the video. So there's 64 different codon combinations. You see that there's redundancy. And so you see that CUU, CUC, CUA, they all code for leucine, so there's some redundancy. We're going to talk a lot about mutations and silent mutations and stuff like that. But just so you know that for translation, they're read in groups of 3. You don't need to write this down yet. I'll bring it back up later. And then this code is universal, so all life forms use this same code. So the transcription unit is the stretch of DNA that's going to code for the RNA. The enzyme that is going to be responsible for this is RNA polymerase. So transcription and translation are a lot easier than DNA replication because you only have one enzyme, which is really nice. So the enzyme is RNA polymerase. All right, uracil replaces thymine. RNA polymerase is going to attach to the region of DNA where transcription is supposed to begin. It's called the promoter. So it attaches to the promoter and it stops at the terminator. So go ahead and pause if you need to keep writing. I'm going to talk about the last 2 steps. Or I'm going to talk about the 3 steps, sorry. So this is transcription. This is the DNA unit that's going to be transcribed. RNA polymerase is the enzyme. Uracil replaces thymine. RNA polymerase attaches to a promoter where it begins transcribing, and it stops at the terminator where it's going to leave and basically stop transcribing. There are 3 steps. So this isn't a bacterial cell. It's very, very simple. 3 steps: initiation in bacteria. RNA polymerase binds directly to the promoter. So RNA polymerase binds directly to the promoter region of DNA to start transcription. That's in bacteria. Okay. In eukaryotes, it's a little bit different. RNA polymerase is going to bind to a sequence called the TATA box. It's a little bit upstream from the promoter. Transcription factors, which are little proteins, are going to recognize the TATA box and bind to it. Then RNA polymerase. So there's an extra step. So in bacteria, RNA polymerase just binds directly to the promoter. Boom, easy. And that's because bacteria doesn't have very much DNA. Mistakes, don't have to worry about mistakes as much. But in eukaryotes, there's a lot more DNA. Things are a lot more complicated, so there's always extra steps. transcription factors bind to the TATA box, RNA polymerase then binds to the promoter area. All right, so pause if you need to. That was step 1, initiation. Step 2 is elongation. That is the same in both prokaryotes and eukaryotes. Elongation is just where RNA polymerase adds the new nucleotides, the complementary nucleotides to the template that match the template DNA. So RNA polymerase is making the mRNA, and it's making it based on what is complementary to the DNA template. So that's called elongation. So pause if you need to, because I'm almost done. So RNA polymerase moves, untwists DNA, rewinds it. So it does everything. That's basically what you need to know with transcriptions. RNA polymerase does everything. Last step is termination. RNA polymerase is going to reach a terminator sequence. Then the mRNA and the polymerase both detach. And so this is the same in both eukaryotes and prokaryotes, except in eukaryotes, this mRNA is not ready yet. It's not ready to leave yet. It's not ready to be translated yet. It's called pre-mRNA in eukaryotes. In prokaryotes, mRNA is ready for use. All right? So that is termination. So pause if you need to. And so this is basically what we just talked about. This is just a review because I'm going to end there. In prokaryotes, transcription, translation both happen in the cytosol. In eukaryotes, transcription happens in the nucleus. And transcription makes this thing called pre-mRNA. So next time I see you, I'm going to talk about what is done with pre-mRNA that makes the mRNA ready to leave the nucleus and then be translated. All right, that's all I got for you. I will start on page 94 next time I see you.